Megan Burton, and my school is Northwestern University. And my poster today is, our goal is to monitor endogenous phosphatase activity using a new assay for profiling endogenous phosphatase activity. So phosphatase is one of the regulators with kinase to regulate the phosphorylation cycle. So the phosphorylation cycle is incredibly important in the body. It plays an important role in almost all cellular processes, including differentiation and metabolism. More than 30% of your proteins use this cycle in your body. Kinases, one of the regulators, has been thoroughly studied, and today a lot of our drugs use kinases as a target and inhibit it. However, phosphatases are comparatively understudied, as they have been proven challenging to study. So we have come up with a new method to study these phosphatases. Old methods, such as radioactive and colorimetric assays, are incompatible with cell lysates because they measure phosphatases that are in the cell, but they don't measure the actual enzyme activity. And phosphatases are all over your cell, so you won't get a signal. And other current methods like fluorescent assays need and require antibodies, which are limited availability. So our assay, which we use SAMD assays, it uses self-assembled monolayers on gold of alkane thiolates. And so here we have our gold with the self-assembled monolators, and then we add our peptides, and then we use mass spectrometry, which is a laser that hits the surface, the molecule shoots off, and then the detector measures how fast it gets to the detector based on its weight, and then it converts it to a graph so you can see. So basically, we created 361 different types of peptides with this sequence here, which don't worry about the sequence, just know that this X position and this Z position represented all the different amino acids, so we could put them on a map and see the different regulation of the phosphatases. So in the central, we'll have a phosphatase works on 3 different types of amino acids, residues: threonine, serine, and tyrosine. So we created 3 different libraries that each had those separate phosphatase phosphorylated residue. So for us, we took our steel gold plate, we put gold on it on 361 spots, then we put our monolayer on it. We then immobilized our different peptides on it, and then all we had to do was add the cell lysate, rinse, dry, and then put it into the machine. And that would show us the activity of the phosphatases, which we then converted to this map so we could visualize all the phosphatases together. So each one of these represents a different peptide that the phosphatase could act on. So you can see these are our results, and what we did was we had undifferentiated cell, and then we differentiated a cell into osteocytes and adipocytes, which is bone and fat cells. And so through the differentiation and the changes in the cell, we subtracted the 2 heat maps to see which ones are regulated. So if it's more red, it means there was more phosphatase activity in the undifferentiated cell. And if it was more green, it means more phosphatase activity in the differentiated cell. So you can see here that there's a lot of different colors, right? If phosphatase activity, or if phosphatases just sort of worked randomly, as in like if they saw a peptide that had something that had a phosphate on it and it would dephosphorylate it, which means catalyze the removal of the phosphate, all of these should be dark or get darker as they move. Or in the same, they should all look similar. However, they don't, which means something's happening. Somehow they're regulated, which is something that we didn't really know today, and we'd only ever hypothesized. In the end, though, our new assay has the potential to provide a better understanding of the mechanisms underlying this phosphatase activity and their roles in differentiation. Phosphatases have the potential to be a therapeutic target to treat many human diseases. And as we use kinases, if we understand how phosphatases work, we can use those to create more therapeutic targets to help treat diseases, all types of human diseases. And that is my project.
Thank you very much.
You're welcome.